Swayam Prabha. Digital India. Educated India.
Welcome, dear students. So we are back with the next part of our Phytosome and Pharmacosomes topic. As we have discussed the various aspects of pharmacosomes and phytosomes, their definitions, their methods of preparations, and basic concept of these lipid complexes of phytoconstituent or the synthetic drugs. Now let us discuss the characterization part in this lecture. Let's start our discussion. After completing this lecture, you will be able to characterize the phytosomes or pharmacosomes, enumerate the methods of characterization. You will be able to correlate the results of characterization and enumerate the applications of phytosomes and pharmacosomes as novel drug systems. As far as the lesson plan is concerned, we will be covering this lecture under the heads of methods of characterization, importance of each method, and correlation of characterization parameters and applications. Let's going back a little bit that phytosomes. What are phytosomes? They're the intermolecular bonding of polyphenolic phytoconstituents with phosphatidylcholine. It proved to be specific and stable, and the resulting molecular complex is more soluble and in lipophilic and organic solvents also. When treated with water, they assume a micellar shape, forming structures which resemble liposomes, but they differ significantly from the liposome, which we have learned in our previous lecture. What were the methods of preparation? Just to recall, solvent evaporation method, supercritical method, which was having SAS method, supercritical anti-solvent precipitation, and the sub methods were GAS, gas anti-solvent, and solution enhanced dispersion by supercritical fluid. And third method was lyophilization method or anhydrous co-solvent lyophilization method. So these were the little bit priming and giving before starting to the characterization part. Now, what were the characterization which we discussed little bit that in drug content, percent drug loading, then SEM surface morphology we will be covering with the SEM, TM, FTR, NMR like spectroscopy techniques, DSC, the thermal analysis will be doing with this DSC and XRPD, X-ray powder diffraction. That is a solid state crystallinity we can assess with that, solubility, permeability, in vitro drug release or permission study, and then in vitro in vivo activity we can address after the in vitro drug release. Now let's start one by one how we can characterize these phytosomes or these pharmacosomes. I'm just giving you some of the examples from our lab that we have developed some pharmacosomes with diclofenac, diclofenac sodium, potassium, potassium salts, aspirin, aclofenac, and so many others. These are just 4 synthetic drugs. Apart from that, we have worked on various plant origin component of phytoconstituent also. So let's see that in case of diclofenac, as we discussed in the preparation method, that acidification of diclofenac salt is mandatory and is a step 1 for moving for the synthesis part, and then in equimolar ratios we complex the drug and the phosphatidylcholine PC80 we took, phosphatidylcholine 80 we took, so a phosphatidylcholine 80% of purity. We got the sample from a major company from Germany, and then the solvent evaporation method was employed and they tried after the vacuum evaporation by applying the vacuum, the evaporation solvent was done, and then the pharmacosomes were obtained. So first of all, going for the FTR spectra, we can see, we can compare the drug, that acidified drug, FTR is PC80 phosphatidylcholine 80s, and that spectra of the diclofenac acid complex, and the spectra of the physical mixture. So we can compare. We will have to take the 4 spectras for each component. If we are saying that for a particular formulation of more formulation with more ratios are there, then you should take the FTR of those ratios also, and then you can compare. Certainly then there is a change in the hydroxyl group range there, that stretching will be there. So you can characterize that what are the changes which are going to be there when you are taking the FTR spectra of the complex, that the liquid lipid complex. DSC, the differential scanning calorimetry. DSC, the thermal analysis, we did that for the acidified diclofenac, and temperature was, we can say, around 170. Peak was, endothermic peak was there, and in 80% PC you can see there the triplet of peak were there, and the, well, in the complex, the thermal analysis, that thermogram was completely different, and there was a removal of peak of the drug in case of complex. So it indicates that the complex has formed there, the complex formation occurred there. So with the thermal analysis we can have an idea whether the complex has been made or not. Okay, had it been the case of in the case of physical mixture, all the peaks were remain there at their respective positions. In case of aspirin also, you can see there was a, you can see that aspirin 80% PC, and then APC 40. You can see APC 80, aspirin phosphatidylcholine 80. This is a complex. You can see the complex thermal analysis, thermal graph, that DSC graph is entirely different, because it is showing the peak at around 50 and 63 degrees Celsius. While the transition peak in the case of aspirin was around 140 degree. So entirely, there is a shift of endothermic peak in the thermal analysis was observed. So we can have an exact idea that what kind of changes were there, whether there were changes or not, whether the complex was formed or not. We can have an idea with the thermal analysis. Now moving to the solid state characteristics. What happens that when we are preparing the complexes, there is a transition of drug from amorphous phase to the crystalline phase to the amorphous phase. Basically, the partial amorphization is occurring there. You can see in acidified diclofenac, it is one of our published paper in the Acta Pharma in 2009. In which diclofenac you can see, and PC80, and the lowest one, the third one in the lower one, this is the complex XRPD, high resolution X-ray diffraction we obtained, that the sharp peaks of sharp crystalline peaks of drug were not present in the diffractogram of the complex, as you can see there. So it indicates that in solid state characteristics there are changes are there, and the drug from crystalline, it is moving to the amorphous phase. Actually in the complex phase the drug amorphization is occurring. Basically this is the turning point. You can say this is the governing point which is going to govern the solubility and the dissolution. Okay, apart from the thermal analysis, so the correlation is desired. So there was a complete shift of peaks, and the peaks were abolished there in the endothermic peaks on the thermal analysis also, while the same is being supported while with this X-ray diffractogram. So the correlation is at most necessary when we are doing the one set of data, when we are comparing one set of parameter with the other set of parameter. Again, in case of aspirin, we can see that APC 80, that aspirin phosphatidylcholine 80 is having, it is again from our lab publication. This complex is clearly showing that the aspirin, which is very crystalline in nature, is becoming amorphized when it is in the complex form, and the complete change and the removal of crystalline sharp peaks are clearly evident here. Okay, again for in the case of aclofenac, which is the analog of diclofenac, you can see the top one is the drugs XRPD, it is again from our publication from Indian Journal of Pharmaceutical Sciences, and phosphatidylcholine XRPD, and then again its complex. You can see that the diffuse peak are there. The crystalline peak have converted into the diffuse peak, and the sharp endothermic, sharp crystalline peaks which were there in the drug are not there in the complex. So it is indicating that there is a change in crystallinity of the drug in the complex when it is in the complex form, when in the phytosome or pharmacosomes form. Okay, for the synthetic drug we call them pharmacosome. Just to remind you. Scanning electron micrograph, like we can also perform SEM. Surface morphology provides a lot of information that what kind of changes are there from the drug to the in comparison of the drug, the carrier, and what are the changes actually in the complex. So you are seeing the SEM of the diclofenac phospholipid complex. So the changes are there in comparison of which we saw the changes as compared to the drug. So there was can say the rough morphology was there, which is indicating that the higher the contact point was there, and higher the contact point when especially it has become in the amphiphilic complex, the higher contact point is indicating the higher solubility, and it is predicted that the solubility will be increasing. So this might be the reason of increase in solubility, supported by the increase in amorphization and the change in thermal analysis. Okay. Again, we can see in acylphenol case, the change is evident there. We can see that the acylphenol phospholipid complex was completely, it is having a very rough surfaces as compared to the lipid and the things. So the surface morphology is a good indicator. Rather, we can also have the particle size analysis through with the SEM, scanning electron microscopy. We can perform for the characterization parameter. Now you might be thinking that how to do these things. You might not be having the instruments in your lab. Here is the solution. UGC DAE CSR. Please note down the website csr.res.in. Our IITs, all our IITs are well equipped with the central instrumentation facilities, and all the IIT students, IIT researchers got all the facility there in house. But the researchers from university don't get the opportunity, or don't get the timely analysis from other labs, and that too they are paid. Okay, so I remember that I had to wait because there is a huge pressure on the IIT labs, IIT instrumentation lab. So I had to wait for NMR of sample analysis 6 months during my PhD. So my PhD was just delayed, around 4 to 5 months submission delay was there, just due to this IIT is queue there, because there is a huge pressure of their in-house pressure is there. So they are it is very typical for them to address each and every one in timely manner. So here is the solution. UGC DACSR, it is a consortium of UGC. Consortium of UGC, it is the autonomous body of Government of India, which is providing a platform to the university researchers, in which DAE, Department of Atomic Energy, has the collaboration with the UGC, and this consortium of scientific research has been made, established, and it has the center in the Indore. The main center is the head office in Indore, and it has got the CSR centers are there in apart from Indore, it is in Mumbai, it is in Calcutta, it is in Kalpakkam. So presently there are 4 centers working. Apart from it, one more center is there. However, it's in accelerator center in Delhi there, but UGC DACSR has the 4 centers there. So it is the website. You can go ahead with this website and avail the facilities. Let's see how. What you have to do, as you can see the scrolling, when you will be putting this website in your browser, you will see that they download user form will be scrolling there. As you can also see in this picture, that the user form, a feedback form is there. Okay, this is the user form. As you can see here, can you see? This is the user form, and this is the link. Okay, this is the link of user form. Now what you have to do, you just have to fill this form, download this form, fill this form, and talk to the concerned scientist which is related to that particular area, and send this form. What is happening that they are having in different centers, they are having the various facilities. In indoor centers you will find the all kind of facilities related to the physics, chemistry, biomedical sciences, and pharmaceutical science, and all other kind of researchers who are involved in experimental research. All the spectroscopy, cryogenics, magnetic labs are there, and thermal labs are there. So a list of instruments available there in there this is available in their website. You can just go there, and depending upon your requirement, you will fill each and everything. What is your sample, what is the nature of your sample, how many numbers of samples are there, and you will have to a discussion, you will have to have a communication with the concerned scientist or concerned person there, and you will address them. Then after the telephonic discussion, you can send this form and get it. The slot will be given to you by this particular center. It may be Indore, Mumbai, Calcutta, or Kalpakkam, depending upon your need. You can go ahead, and a date slot is given there. You can perform the experiments there. Just you will have to take the samples there. You cannot send the sample by speed post or by post there. You will have to go there, experiment, do the experiments, get the analysis done within 4 5 days, 3 4 days. You can go there, depending upon your number of samples. You will be provided, rather the students, research scholars are provided with the hostel facilities, paid hostel facilities, and the TA is paid. TA is paid for the sleeper class fare is given to the research scholars for coming and going to the centers, and it's wonderful and free of cost. All the analysis, there is no analysis fees, and you will have to have a discussion after the experiments. You will have to submit a report after you reach to your destination, and this is very beautiful experience, very nice, very wonderful experience, talking and interacting with these scientists. I am working with these scientists, the hardcore scientists, I can say. They are wonderful. They are doing wonderful work for the sciences, and they are very collaborative and very cooperative, very supportive. And I have been fortunate enough to have the interaction since 2003 with them, with I think from Indore Center specifically, and after that Mumbai Center, Professor Siruguri, and from Indore Center, Professor Sinha, and from Calcutta Center, Professor Saha, Dr. A.K. Saha. So all these are very good and very supportive for your work. Please go ahead and avail this facility. I'm moving ahead now in our discussion. Now solubility is the thing. Like solubility, there is a 2 to 10 fold increase in aqueous solubility in general, and while the permeability is also increased, but not to a greater extent. Solubility, you know how to determine the solubility. I am not going to the detail. Now we move to the dissolution study. What you have to do from the dissolution apparatus, you will take and you will compare your drug release in comparison to the plain drug. You will compare the drug release of your complex, prepared complex, and you can do the kinetic studies, and model fitting you can do depending upon your requirement. As you can see also here, in the case of aspirin, the lipid complex showed a better increase in dissolution profile when it is in complex form, or you can say in pharmacosomes form. Okay. Now, as far as the phytosomes are concerned, from our lab we have prepared a a good number of, I think, a total phytosome and pharmacosome we have worked upon 24 different drugs. And among the phytosomes, gallic acid, naringenin, catechin, quercetin, and I think you name it, we have done all the work on almost 13 to 15 phytoconstant, imodin, catechin, apicatechin. Okay, we have worked on these, and we have got very wonderful results. Now moving to the whether the component is phyto constant or the synthetic one, your characterization parameters are going to be same. Now other physical evaluation of phytosomes or pharmacosome, maybe carbon 13 NMR you can perform. You will see the complete shift. You can see the complete shift provided you are having the good purity phosphatidylcholine, because it is my own experience that if you don't use a high quality or high purity phosphatidylcholine, you won't get a good NMR. So it would be very typical if you are dealing with the 40% phosphatidylcholine. You won't get the NMR. You will get the NMR obviously, but you won't be able to interpret the data. So it's better to deal with when go for NMR only when you are having the purest grade of the phosphatidylcholine apart from the drug, apart from the API. Proton NMR you can also do. This is the work from the Indianas people, Indianas scientist team, which is working on the phytosomes. Okay, it is for catechin. I am just showing from their website. It is there. It is not our work. In vivo performance, Ginkgo. I have been not I was not fortunate enough to get the in vivo done because of lack of facility. But if you have the facility of in vivo, you can you can compare the plasma level of phosphatidylcholine complex or the phytosomes in comparison to the drug. As you can see that this upper one data is for phytosomes and this lower one is for ginkgo biloba extract. So there is a clear cut interaction, clear cut change in the bioavailability with the phytosomes. So good increase in bioavailability is there. As we can see, this area under curve is very good in case of area under curve is very good in case of. And same in vivo performance of silibin, you can also see in silibin phytosome, you can see this is the silibin, blue category. In the left one, you can see the silibin is there, and it is the upper one is silibin phytosome. Okay, so you can see these phytosomes there, and they are having a good bioavailability, good area under curve, so indicating the higher bioavailability with case of in case of phytosomes. So we have got good results with these uh, lipid complexes in case of phytophospholipid complexes. Many times they are called phytophospholipid complexes, phytolipid complexes, or simply lipid complexes. So just go ahead, good number of articles you will, good number of studies you will find to address your query how to do these in vivo studies. However, these in vivo studies depend upon which kind of drug you are taking, and not only plasma concentration, you can also see the effect depending upon the hepatoprotective model, anti-hepatotoxic model you can take, and paracetamol induced hepatotoxicity or other models depending upon your need you can take, and you can see the therapeutic effect there in the as part of the in vivo study. Now, what we can do for improving the effectiveness of phospholipid complexion, the use of advanced new techniques like supercritical fluid and live flashing method can be employed for this kind of improvement in effectiveness. Moreover, the use of high purity grades of phospholipids with advanced and effective processing and packaging, the product may result in stable liquid complexes with better shelf life. So the stability is also, because whenever there is a lipid, there is an issue of stability. So what you have to do, you need good purity grades, good processing and packaging techniques. Then only you will be able to produce a stable and effective product. Now moving to the application part of the phytosomes. When we are discussing the application of phytosomes or pharmacosomes, uh, first and foremost thing is it is used for improving the biopharmaceutical properties, whether it is solubility, permeability, okay, partition coefficient, you can see. And we can directly address these things. We can directly, with the in vitro studies, we can perform the characterization parameters for poorly soluble drug, for poorly permeable drug. They can be used for improving their biopharmaceutical properties. Second is improving the bioavailability, as we have discussed that when drugs are being complex, especially the phytoconstituents are being complex with the lipids, their bioavailability is improving quite many folds, to 2 to 10 folds increase in bioavailability is there. Okay, and which is in good tune with the increase in solubility or dissolution. Okay, then as far as the therapeutic effects are concerned, for better cardiovascular effects, for better anti-inflammatory, anti-skin aging effect, antioxidant effect, anti-hepatoprotective effect, anti-cancer effect, the lipid complexes show the better therapeutic effect as compared to the parent drug, especially with respect to the bioavailability one, and in some cases, underline, and in some cases the therapeutic effect is improved due to synergistic effect of the phosphatidylcholine maybe. However, the main purpose, the main application is improving the biopharmaceutical properties along with the bioavailability of the uh, drug under concentration, or the API for which you have prepared this uh, lipid complex, either in the form of phytosomes or in the pharmacosomes. Okay, these are some of the references which we have published, and some other references are there. You can go ahead. Lot many things are there. Now let us summarize. Let us summarize. Phytosomes are phytolipid complexes. Okay. Pharmacosomes are synthetic drug lipid complexes. Basically, they are drug phospholipid complexes. Phospholipid may be of any type. There are several types of phospholipid. We have not gone into the detail. We have not taken you into the detail of phospholipids. You can take depending upon the availability, because it is not very easy to get the good grades of purity in the cost effective way. Okay, I was fortunate enough to get the free sample from a company, Lipoid, but you have to focus on the availability part also first. Then what you get, you get the improved biopharmaceutical properties, you get the improved bioavailability when you are preparing the phytophospholipid complexes or the synthetic drugs phospholipid complexes. As far as the characterization part is concerned, you do the characterization for the surface morphology with the SEM, TEM, or maybe other parameters may be taken. Solid state X-ray forward diffraction you can perform, and spectroscopy you can perform. Spectroscopy you can perform like proton FTIR, you can do your NMR, you can do. Okay, and obviously you perform the UV for the drug content. Solubility, you will have to perform. The drug release is done in the dissolution apparatus standards, and the in vivo studies can be done depending upon the various pharmacological models, where depending upon your need you can adopt any pharmacological models which fit into your criteria of drug and its therapeutic effect. You can check it out and play a very vital role in efficient drug delivery with respect to the bioavailability and biopharmaceutical properties in particular. As far as the applications are concerned, apart from the biopharmaceutical properties increase and the bioavailability increase, the improvement in cardiovascular, anti-inflammatory, hepatoprotective, anti-hepatotoxic, and anti-cancer activities are the major applications. Various studies have been done on improvement of these effects by the phospholipid complexes of the synthetic drugs or the phytoconstituents. So it was all about the phospholipid complexes. We have given an overview about your how you can perform these studies, because you know university, it might be the case the university or the lab may not have the all these equipments in your in your household laboratories or in your household conditions. What you can say, the in-house experimentation is not possible for each and every parameter, so you have to go for the sampling done for analysis. So you can go for the UGCDE consortium. UGCDECSR is doing wonderful job and serving the nation, serving the university, academia, researchers. So avail this facility. This is my humble request to all the researchers. With these words, I would like to conclude my lecture on the phytosomes. We'll be coming back with some more lectures on the node ready system in the coming weeks. Thank you. Thank you for being with us. Happy learning.